minutes.
Oh, sorry, I forgot that. Just let me then. Hmm. Okay. Welcome to the second seminar in our biology speaker series. Today's seminars, A Journey to Microbiology in Space, will be presented by Dr. Seragna Guadarrama Ventral. She was born and raised in the Estado de Mexico, Mexico. She's a proud first-generation graduate with a non-traditional upbringing. She earned her PhD in microbiology from University of Dublin, Trinity College, and Ireland in 2013. She's associate professor at UTRGV and School of Medicine. In this presentation, she will discuss her academic training and overall professional journey. Additionally, she will explore the effects of the space environment on life, including how microbes survive and thrive in space, the challenges, opportunities that space life and microbiology present for virulence and drug resistance, and the implication of this factor for life on Earth and space, crude both past and present. Please join us and welcome Dr. Guadarrama for this fascinating exploration on microbiology and space. The floor is yours.
Thank you. Thank you for your kind introduction. Thank you. Good afternoon, everyone. I'm not sure how many of you are here. I cannot quite see you, but I hope it's a really good turnout. I made sure that I invited many people to this talk, not only because we're going to talk about space microbiology, but also because I like to reach as many people as possible for whomever is interested in joining the space program. All right, so without any further ado, let's dive into space microbiology. So let me get started with something that you might be familiar with. Maybe, right? Some of you, maybe not. This is from the movie Alien from the 1970s. Gen X, pretty much familiar with it. So we are here to talk a little bit about how that could be quite possible, right? That could not be so sci-fi. It doesn't have to really be that scary because many of our microbes might look like that, but they are not that big, thankfully. So, okay, so how do we get started? How do we can actually have in mind that this is quite possible for life to thrive in space? So that's why we have this research on space microbiology. So just to give you an idea, right now, up there in the International Space Station, there has been some discovery as far as microbes are concerned. They're called novel bacteria, right? This was published in 2023, and these are just a few of them. Okay, as you can see in these micrographs, they're quite cute. That's how I refer to my microbes in my classes, but we see some of them, Anthropobacter, Lifusonia, Sporacina, and all these different ones. I mean, it's not to say that, you cannot find something similar here on Earth, of course, but I'm just saying that these are novel because apparently, there's some evolution happening too up there. Okay, so that's that up there. Now, there's been a lot of emerging research as how to study microbiology here on Earth. How do microbes can react, or how do they react, response to the environment of space. So for that, we have this different equipment. Now, back in 2005, I did research on this field, and I tried to assess bacterial virulence. In other words, the capacity to infect of a particular bacterium. And I use what we know as model microgravity. And this is the machine that is used. This is just like a general type of example of it, and I'll explain more about that in the slides to come. All right. Now, here my disclosure is that I don't have any financial interest to disclose this point. And so what am I going to be talking about in the next hour or so? First, I will talk about what got me into this field, my education, work, and different activities that I've done throughout my career, my academic journey, my professional journey. And also, that will be the next step on that would be to talk about microbes in space, right? The significance of it. What does the space environment tell us about supporting or sustaining, right, microbial life in space? And so to the survival of microbes, right? We'll talk about space flight, microgravity, back in the day when we had that Mars meteorite chunk, what do we learn from that? And the challenges that microbes encounter in order to thrive in space. And then we will conclude with talking about a little bit more about that capacity to infect with microbes and the drug resistance that they evolve. How does the crew health, how is it affected by this? The present challenges, and how can we circumvent or how can we combat the antibiotic resistance developed by these microbes up in space? And where is the space microbiology today? All right. So what got me into this? Well, I have to say, back in 1993, I wrote a handwritten letter to Dr. Carol Sagan, and because I was pretty much lost as to where to go, what career to follow, what would be my path, right? Like as many of us are when we are very young and we don't know where to go. I mean, we have passions, we have hobbies, and we don't know where to go. So my parents encouraged me to write this letter, and they say, just write to Dr. Carol Sagan, because since I was a very little girl, I've been following everything about him, everything that he has created, is, beginning with the Cosmos, the series back in the 80s. And so they just encouraged me to do that. And I was like, are you sure? I mean, I don't know if he's gonna, really pay attention to me. I mean, who am I, right? And so my parents said, no, it's not about like, who are you? I mean, yes, who are you in a way, because all the things that you want to do, and he's the perfect person to ask. So I went ahead and followed their advice. And to my surprise, I got his response. My parents tried not to be too surprised because they were pretty sure he was going to answer. They had this vision, they say, and so this was his response. And so something that I want to point out is that he said, for those of us who want to embark into this field of the origin of life, right, investigating here on Earth and beyond Earth, and also looking for extraterrestrial life, because that's no longer a sci-fi type of phenomenon. It's something that it's serious and it's worth our study, our science and technology, right? So he suggested that I work in an established field, as you can see here, perhaps involving microbiology, biochemistry, and molecular biology. So he was very wise to tell me that so that I could have those marketable skills if we do not find extraterrestrial life, right? During my lifetime, that is. And then if we do, I would be prepared at a fundamental level. So this is just something that I wanted to share with you again, because it's very important that we have that always in mind, that no matter what our dreams are, they can always be attainable, and they are, we can make them a reality. We can make them shape the reality we live in, right? Okay, so just a little bit of a context about Dr. Carl Sagan for those of you who might not know him. He was the one, besides the creator of the series Cosmos, which I highly recommend for you to watch if you haven't done so. He wrote the novel Contact Jodie Foster. He made the movie Jodie Foster, and then he wrote Murmurs of the Earth, which was based on the Voyager spacecraft, which launched back in the 70s, and it was carrying a plaque. When I talk to people about this, they think I'm talking about a movie. No, but this is quite real. In the spacecraft, there's this plaque, the Voyager Golden Record, which everything about human civilization is in. So, like right now, is beyond the solar system. And so the objective of that had been to, if there is extraterrestrial life out there, intelligent life, right? If they come in contact with this plaque, they would know that there's humans in the solar system. So it's very interesting. And then in his words, the spacecraft will be encountered and the record played only if they're advanced a spacefaring civilization in interstellar space. There's music, there's everything about us. So it's quite amazing. All right. So education, what I've done so far to find me where I am right now, I have a doctorate in microbiology from Trinity College Dublin in Ireland. I have a Master of Science in microbiology from Montana State University, and another Master of Science in cellular biology from the University of Alabama in Huntsville. And my bachelor's is in microbiology and chemistry, and that is from, and space science, of course, and that would be from Florida Institute of Technology and the University of Texas, El Paso. So from all these, I really obtained precious and valuable information as to where do microbes stand on this earth and what are they capable of, also in extreme environments, which we will be talking about. I was very fortunate to be able to go to these different places. I'm very thankful, very appreciative of the people I work with, because in every single place I had the fortune to work, to also pay my own tuition. And not only that, but to have the experience to work with NASA. With the exception of in Trinity College, I was more in touch with the European Space Agency. All right. And then as far as work is concerned, so I had worked in all these different places where I had studied, as I had mentioned before. And besides what I had mentioned, in 2013 to 2014, I had worked in Singapore, and I was a science communicator there. And it opened up my eyes to other types of thinking, right? And other types of making science, and how biofilm, the actual field of biofilm, microbial biofilm, does in the world too, because it's not only what happens to microbes in space, which we will talk about biofilms, but also what does that mean here on Earth. And so something important that I have to say that made much of an impact in my career, in my academic journey, in my professional journey, is that I had worked also with NASA as a Space Life Sciences Flight Counselor back in 2001, 2002. And I got to work with astronauts back then, and I'll show you a few pictures in the slides to come. And also back in 1998, I had the fortune to work with Dr. Marion Lewis. And during that experience, so she showed me how to set up a payload of experiments, a sort of experiments with Jurkat cells, so that we could study the effects of a spaceflight on these mammalian cells. This payload was managed up in space by veteran astronaut John Glenn, who back then was a senator. He passed away, unfortunately, but he was well in his 70s when he flew back in 1998 in pretty good condition. So that's just to show that age is not really, it's just a number, right? So again, whatever the dream might be, the age might not be really an excuse, right? And what else then? 2003, 2006, that I was also involved in space science, so to speak. I will talk about that too. And that one research was sponsored by NASA and also the European Space Agency. All right, I'll talk about my work in Trinity College, Montana State University, and the University of Alabama a little bit. So in the slides to come. Okay. So something I do want to, I can't emphasize enough, and that I learned along my professional journey, is that it's very important that you surround yourself with people who fight for you, okay? In those places where you are not in necessarily, right? And that's just very valuable. And I'm talking about allies, right? In having allies around you, those alliances, and more so for us women, right? In this day and age, and the fact that I'm a minority, and I'm sure that the people who are attending this seminar uh, belong to minorities as well. So this is very important. Okay, so this is the activities I've been involved with. This is back in the 2000s when I was working in NASA, and I was inside the cockpit of Space Shuttle Discovery. We were sampling in the insides to understand, if there was a different microbiome. Back then, we didn't really understood it as a microbiome, but now, we have much more knowledge on that, and I'll speak about that later. But those were the beginnings, right? And also I had the fortune to work with Dr. Chang Diaz. He's from Costa Rica. He's a retired astronaut. He has his own business now, and since he's an astrophysicist. But it was really a pleasure working with him, very kind person. And yeah, that was back in the 2001. Now, forward time, and I'm here in the School of Medicine, in UTRGV School of Medicine, and I talk from time to time to medical students or to the community at large about what is it to have an involvement in the space program, particularly as far as microbes are concerned, right? And then they're amazed by this. I also talk to middle schoolers, high schoolers, and it's important to spread the word. Here's some more pictures with the team, sampling pre and post flight back in 2001. And please do notice our attire here. It is very important to be like this, and just keep that thought. That's something that I will come back to. So that's quite important. And maybe you can tell me, I don't know how much you can participate here with me, but this is something that I usually ask the audience, like, why would you think that I have this attire? Or why would the team would be wearing this attire? Okay, so for those of us who have thought that we are in control or we are at the top of the pyramid here, we are not. Actually, microorganisms are. And Neil deGrasse Tyson couldn't be any more right than in this quote by saying that in 1 linear centimeter of our lower colon, okay, there lives more bacteria, about 100 billion, than all the humans who have ever been born. Okay, yet many people continue to assert that it is us, okay, who are the ones in control or in charge of the world. That couldn't be further from the truth. It is the microbes, because they were before us and they will be here long before after us, after we have extinguished. All right. So what is the significance of microbiology for NASA or for the space industry? It is huge for many different reasons. So these are this encompasses pretty much the NASA Office of the Chief Health and Medical Officer. Like they listed all the different things that they do regarding health, so to speak. So for one, the microbiome is extremely important to protect the health of the space crew. There's also the environmental levels around, right? Like the actual conditions of the rocket ship inside, like the spacecraft, or the airlocks, the insides, the modules of the International Space Station. There's also the treatment, the management, right, of those astronauts in flight, but not only in flight, pre and post flight. Oops. And all these all together, which those are the three main components for space microbiology, they come hand in hand or are included in what is known as aerospace medicine, right? So aerospace medicine is like an umbrella term for many different factors that had to do with space life sciences. Okay, so in this slide, all I want you to think of is that what do we see here, right? You would say, oh, well, I see a virus, I see a bacterium, I see some type of microbe, or for those of us who got really traumatized by the pandemic might just say, okay, oh, well, that's SARS, the culprit for our pandemic, SARS-CoV-19. Well, yeah, it looks pretty much similar. But I want you to keep this picture in mind and then look at this, right? So we are all one. This might get a little bit philosophical, but it's important to understand that we share a biosphere, right? And microbes are here with us to help us thrive on this planet. But as they help us thrive on this planet, they actually carry the clues for us to also go beyond this planet. Okay, so with that in mind, think of this. Okay, if it's that, if that's a fact, if we take that for a fact, then there's no use for us. It would be unthinkable to make them go expire, right? Like many people think, like, let's just I don't know, extinguish such and such bacterium or such and such virus because it's no good. No, every single microbe is important, just like any single other living organism. And that's only because this biosphere, okay, has an evolution record. It's been there with us. It has somehow helped us thrive on this earth, right? And the record of it is 3.5 billion years. It has evolved, it has developed an atmosphere with pressure. There's temperature just right for us to be here, to develop life. It protects us. Okay, we all know this, right? But not only that, there's gravity too. Otherwise, we will be flying off, right, up outside Earth, as much as many of us might want to have that, and just kind of drift off to space. But we have this constant acceleration, right, of gravity pulling us down to Earth. That's 1G. Okay. So having said that, think of it like we have this atmosphere on Earth, and then we needed to have something else in order for life to continue evolving, right? So that it could emerge and then could develop and it could thrive, because we had the many different other things, even though I had said that we have this biosphere formed. But in order for that biosphere to form and protect us from UV light, for example, other things might have to happen, right? So there's these thinking, one of the theories, right, would be for the origin of life, that there was this meteorite delivering the building blocks of life, despite all this UV radiation. But then it's like as if Earth was preparing for it, right? And then we have like this cosmic situation, so to speak, and the primordial soup with all these different chemicals, that it was just right for life to emerge. So what am I talking about? I'm talking about that if we have UV light, there's got to be just the right conditions for it, for the organisms to be effective, right? Not only to appear, but to be able to reproduce, to develop, to evolve. And so everything was just right. And it was, from early Earth all the way to present Earth, the conditions were transforming. So we've got all these, right? So for the first organisms to appear 2.1 billion years ago, right? Moving on. And then we have all these different things that make up the now biosphere, right? So the ozone layer helping us to counteract those UV lights that might be harmful to life in itself. There's oxygen from photosynthesis, and this can only be true because there are microbes that are doing that for us in the primitive Earth, right? And then finally, life can go on. So talking about this space environment a bit more, especially for the Earth's atmosphere, what happens? There's not only one layer, there's different ones, right? We have that something to remember is that here, the first layer is the troposphere, But then look, there's about what, 1, 2, 3, the third layer up here, about 100 maximum here kilometers in altitude. This is the limit where bacteria have been found. So this is another question that I would ask my audience. So do you think that you could actually find microbes being resilient enough? Would there be one resilient enough to withstand anything above that? So I would have some people say yes, I would have some people say no. Most people would say no, right? But you'll be surprised how resilient microbes can be. And I'll be making some comments here and there as we move along. So what do we know about life in space? So let's get into business here, right? So what we know in a nutshell is that we understand at least the tip of the iceberg, the microbial behavior up there, that there is a containment vessel that we're dealing with, that our immune system might not be in the ideal conditions. And then the antibiotics and infection treatments are not as effective as on Earth. So there are many different things that are not quite as expected. And you know, talking about the microbial behavior here, what does this mean? That increased capacity to infect, that's what virulence is. And so there's been several different microbes that have grown much better in the microgravity environment or space environment that here on Earth, just like as if they're like remembering where they came from, you know? And that's what I'm saying, right? That they're giving us clues where we came from, perhaps, right? And then the microflora exchange, meaning like the microbiome exchange happening in your body, the antibiotic resistance, all these is increased. And latent reactivation, meaning that, you know, for those microbes that go dormant in your body, for example, herpes, which is a virus, might become reactivated being up there. But that's also, it just makes sense because your immune system, right, is weakened being in space. Okay. So, so would it be possible for life to survive outside the Earth's atmosphere? That's the other question. So that's what we're trying to understand. There's many things that we have attained and that we need to understand the physics of it. Like if you look into this image from a paper published by Cheryl Nickerson in 2022, so we have Earth, right? We have the gravity of Earth at 1G, then LEO, which is low Earth orbit, and most of the experiments that are being done in the International Space Station are actually, there's a parenthesis or somewhere there's a footnote saying that they were done in LEO, low Earth orbit, which is very different as to if we're talking about somewhere interplanetary space. It's a different gravitational condition, right? Because this is, like I said, microgravity, and probably out here, maybe interplanetary, but maybe much even beyond, like interstellar space, that could be zero gravity, because there's still some pull, right? Like some gravitational pull here, and that's why we call it microgravity. And then, you know, as we're moving right up along here in this, like in this image, I mean, ultimately, we want to go to Mars and see what are our hopes, what is the possibility, the potential for life to thrive there too. So that's why we're doing all these, and we can consider all these different things in which we don't have, right? Microbes don't have, and if that would be quite a challenge for them, because it's like we're pushing them first and see what happens. And then how can we use them to our own advantage to help us, to guide us, you know, as we embark to space, right? So here we go again, right? Like the challenges, what does it mean, the space flight? What does it mean, the microgravity that we'll be exposed to? And also I will be talking a little bit about this Mars meteorite. I think I mentioned it before, to understand if indeed microbes, you know, really can thrive out there. So what do you think happens to microbes when they're exposed to altered gravity? Say space flight, because that's one thing, and microgravity, this is another question that I would pose my audience. So there would be different people participating, saying different things. And so here we ask another question. Are microbes and humans different from each other? Because it's important for us to draw that comparison, right? Some people might say yes, some people might say no. We're living organisms and we share many different things. You'll be surprised. When we are faced with challenges, what do? Right? We can either become aggressive or we can just freeze the fight or flight situation, right? We're not very different. So microbes, as it happens, when they are exposed to these spaceflight conditions, they become more virulent, more aggressive, which means they infect much more. And that's only because it's a new environment, right? And then also they have exhibited more antibiotic resistance. And then not only that, the growth dynamics, okay? The way they grow is also changed. And this is all in response because the environment has been changed. Okay, and this is all because they want somehow to thrive, and so there's different genes that are being expressed. All in one word, they want to adapt, and so they do it beautifully and very elegantly. Adapt, that is the word. So what does it mean for a microbe to adapt? What does that really mean? So if there's an altered gravity environment, like we have mentioned, it could be that LEO, which is in the International Space Station, right? Or the space flight during launch conditions and they're coming back. They need to adapt. Perhaps there might be that less nutrient availability. They have to get ready for that in case there is. There might be movement or no movement, right? There might be growth limitations, and there would be that fluid shear decrease. I don't know if you're familiar with this word shear, but when you encounter space conditions, shear, is just that fluid dynamics, right? The fluid dynamics, if it's increased or if it's decreased, is it better? Is there much fluidity or is it not as much? What does that mean? And so the membrane could change, the membranes of organisms, depending on how the fluid fluctuates, right? But as it turns out, in those space conditions, it decreases the shear. Okay, so what do? They form biofilms, and like I mentioned before, this is kind of like an emerging field too. There's not extensive yet research on biofilms, so that's something that you may want to look into if you want to embark into the space life science research, because it is really fascinating how biofilms work and the reason of their existence, right? So microbes construct biofilms. So what it is? So for example, in the space station, there's the problem of the water pipes. So I couldn't find a better one like this image from the water industry in Australia, which they are showing how this biofilm formation is a problem in the pipes, right? And also in the space station, as it turns out, for water. But so they do this, and if you think it's a problem here on Earth, it's even worse in space station because, like I said, the shear is decreased. There's not as much speed of water, right? And so there's that tendency of this film of bacteria, in this case, to form. And it's really viscous and it's really sturdy. And they do this for different reasons, right? Because once they do that, they want to be able to harness in a better way those nutrients. They want to seize those new conditions, so to speak. They're better for them. And then they can better get everyone together, because it's not only by one bacterium or one type of microbe. It could be polymicrobial. Different microbes could be actually in this type of environment. And so when they're trying to communicate, just like when we are in different meetings, we say, we have reached quorum. Well, guess what? Microbes do the same and they have reached quorum. In biofilms, it's much easier. And so, so that they can move along, or they can actually communicate genes, because that's their language, through genes, and acquire resistance to antibiotics. Who knows, right? And just like I said in that other bullet, resist the attack of antibiotics and reorganize the whole genetic structure in a faster way. So for this reason, we need to safeguard absolutely the astronaut crew, because microbes are ahead of us. So we really need to catch up. And then, people have said, okay, well, maybe, when you send those spacecraft or the space probes or whatnot out there, maybe it's overkilling it too, right? Because there's no such thing as 100% sterile. It's just impossible. And that's quite all right. I mean, we cannot really be in such perfectionism, and also because life finds a way anyway, right? We know it. And like I said, here also we're walking microbial universes. We have different microbiomes in our body. So as we try to sterilize this equipment, like in the space station, it's going to be all over the place. So let's think for a moment like a microbe to understand this process of adaptation, because this might help us out too as we're trying to adapt to the conditions of space. So the 3 R's, I call it, whenever I talk about resilience in microbiology, and how, different aspects of disease evolution and the such, is that there has to be the right numbers. There's got to be the right time, okay? Because this is also going to tell you if a microbe is going to stay commensal, which is really going to do nothing to you, or perhaps help you, or becoming pathogenic, okay? So it has to do with the numbers, the right time, and the right place, right, for that microbe. And so all these 3 are fundamental for them to establish and thrive in any given environment, no matter what, okay? Because remember, they have to face very difficult situations out there: isolation, radiation, microgravity, hostile environment, distance from Earth, right? It's just, to say they're going to go through some, hardship, but not for too long. So, because they have been trained much like far away before us, right? They have been trained here on Earth. They're extremophiles, guys, okay? And I'm sure that you have heard about them. And so these extremophiles you can find in many different places here on Earth, okay? It's like, I don't know if you have had the chance to watch any videos, any documentaries, or if you have been fortunate enough to be in any field trips going to any of these places. But I can tell you that when I was doing my master's in Montana, I had these field trips to Yellowstone Park, and I just couldn't close my mouth. Every time I was there, I was in awe all the time. Just, it was like going back to primitive Earth, okay? Because, being witness to all these different microbes trying to withstand extreme conditions here on Earth is just the same as looking at them in any other places here in the solar system, for example, right? They can survive extreme conditions. I'm talking about low temperatures, low pHs, high pHs, salty acidic condition, that is, you name it, right? When I'm talking about hot springs, that's, by the way, in Yellowstone Park. Or not only that, because this is Yellowstone Park, by the way, sorry, I forgot that I had that image. But look at the size, the dimension of this. This is almost 120 Fahrenheit degree type of hot spring. And then you might be asking, okay, how is this possible, right? Like, how do you attain all these colors? Some of you might think this is AI, but this is not AI. This is quite real. And like I said, I was fortunate enough to be there a couple of times, or more. I don't kind of remember, because I would just be very amazed every time I would go. And it's extremely hot. I mean, of course, not, you cannot go in there, but look at the people that are working there, and you could feel it, how hot it is even at that distance. And this is quite possible because this is full of cyanobacteria. and they can withstand very high temperatures. And then their metabolites, going through these different photosynthetic pathways and processes give us these beautiful colors. And this is primitive Earth right here for us. But like I said before, it's not only the temperatures that they can withstand. They can even go from hot temperatures, that would be one extreme, to even gamma rays. That's another one. You wouldn't even believe it. Remember Chernobyl? Maybe no, because I guess I'm old already, right? You are not old enough to remember this incident, but back in the 80s, 1986 to be precise, there was this huge explosion in Russia, right, in Chernobyl, and they had all these different kinds of radiation happening, and you would think no life could actually, survive that or thrive right after that, right? No, but there's, life finds a way, right? And so, even after, with all, it looks like a science fiction movie, right? But this is quite real. And so we have what we call Thermococcus gamma tolerance. Yes, there is a bacterium. This is a non-pathogenic bacterium, but it's not that you got it from Chernobyl. No, I'm just saying here, just to give you an example, that there is microbes that can withstand, just to give you context, big amounts of radiation, in this case, gamma rays. And this particular microbe was isolated from the Guaymas Basin. Okay, hydrothermal chimneys, remember I had mentioned that is one kind of an extreme condition. And so this strain in particular had to be enriched, the culture had to be enriched with gamma rays, massive doses of gamma rays, like about 30 kilograys. Now, what does that mean? To give you some idea, the Department of Agriculture in Australia, they have said in one of their in their policies that they needed to irradiate for biosafety procedures about 25 kilograys, right, to make sure that there was no life, like they could sterilize things. But now, guess what? Given all this information about microbes that can withstand that, they had to raise that, right? I think up to 55 kilograys to make sure that they could sterilize equipment and such. But at this rate, there's more extremophiles. I don't think that, like I said, that we can reach 100% sterility. Okay, but this is so important. This is so fascinating because studies have revealed radio-resistance proteins, so to speak, right? In a way, because there's, like I said, life finds a way. So DNA would find a way to repair any type of mutation and the such in life with evolution. And so I am mentioning all this because, remember, we're moving into space conditions. So back in 1996, what happened? There was a very controversial thing going on. Remember, that was back when Bill Clinton was here in the US. He was president of the US. So the Mars and the meteorite hunters discovered this meteorite back in 1984. And so they said that there was possibility of life outside Earth, as they found these in the meteorite that had been found in Antarctica, right? And because they saw these forms, as you can see here in this image, and it was even photographed, and it was a big thing, right? They said that what they found to be probably just looked very like the size and the textures of some of terrestrial bacteria. And then, that they could pretty good be fossil remains of a past Martian biota. But then, you have to be objective as a scientist. So thankfully, back then, the US President Bill Clinton, he said, we need to really examine this and we need to let other scientists look into this. We cannot just really jump into conclusions. And that's what happened, right? We got very excited about it, but we needed to remain objective. And then later on, 2022, there were more articles published on studies like this. And so they reached the conclusion that that this is purely geological structures that could mimic organic structures. So who knows? They could be just skeletons, right? that could be preceding something else later on. But this happens more often than not in geology. And so geologists call it chemical gardens, right? So it was, sadly enough, it's not like we have found life on Mars or or some type of clue of the origin of life, or maybe not, but we know we can continue studying and understand what's happening. So the challenges that we have encountered with spaceflight and microgravity are several, right? Just like I have mentioned, radiation and all these, and then there's increased virulence. Some of the bacteria, some examples of that is Pseudomonas aeruginosa, Salmonella, Streptococci. And there's also what's been found to have decreased, which is Yersinia and Staph aureus. There's biofilm formation, as I have mentioned. This microbiome changes, like Clostridium difficile, lactobacilli, which is very important for us women because it inhabits the vagina and it helps us in many ways not to get ill down there, right? And so all these, like I had said before, also there's reactivation like herpes viruses. So there's many different challenges which the microbes have encountered, but they have come through somehow, right? This is just to show you, as far as fungi, there's been many of them that have been found in the space station. There's many different bacteria that have been found too. And just to give you an idea, fun fact is firmicutes and bacteroides are found in the colon. And so these were, come from a study from those particles that were collected with different equipment to study the microbiome in the space station back in 2015. So let's have a look into how to study that virulence and the drug resistance up there, what's happening. So let's recap. Spaceflight conditions. We know that growth in bacteria, in microbes in general, is enhanced. Antimicrobial susceptibility is altered, meaning that they are not as sensitive as on Earth necessarily. And this affects the virulence of microbes, right? The capacity to infect. So we also know too that astronauts experience secondary immunosuppression because they're no longer on Earth, right? And so they're going through so many changes, and so the body is all confused. And so if you put all this together, what does that mean? Recipe for disaster. Because if that's the case, right, what's going to happen? Astronauts are going to be much more vulnerable for disease and infection because of this increased virulence in bacteria, microbes in general. So we need to study more microbes, right, in this context of space. So there are limited opportunities, though, right, for space flight, and so we need ground-based simulations. So this is where we have model microgravity. It comes to the rescue. This is something that students, in particular my medical students, they find it extremely interesting. I have a few engineering students from the College of Engineering who are currently working on a device that we could use for better studies in space microbiology. So what does that mean? That these equipment can actually simulate microgravity conditions, or it could also be called low shear microgravity. And also Nickerson, Dr. Nickerson refers us to the rotating wall vessel because it rotates, and I will show you in a minute. But so how do you achieve this, right? So this is two different scenarios. You have the simulated microgravity in which the axis of rotation, here this axis, right, of this, oopsies, it's perpendicular to the gravity vector. So that's considered simulation of microgravity. It's perpendicular, that axis, to the gravity vector, and this will attain or will achieve low shear fluid dynamics. Whereas for the control position, which would be the Earth gravity conditions, we would have this, which is parallel to the axis. The gravity vector will be parallel to the axis of this rotator. So keep that in mind for what's next. So what does that mean for the bacterium if we're studying the bacterium in this type of equipment? So we are trying for the bacterium to think that they're in space, right? And so they will find nutrients readily, much more easier, much more easy than than on Earth, right? Like at the 1G control, because they're all at bottom because gravity is pulling them down. And so they're all happy here, spring break, yay. And so they're getting all the nutrients here, right? So this is all really good for them. So we're trying, just like I said, simulate those space conditions in this model microgravity bioreactors. They're also called the HARPs. This is a name that one of the companies here in Houston, Synthecon, has given to this type of vessels, the HARPs. And so they have shown to be really useful. And another thing I want to mention really quick is that they attain free fall. Like when you have a bucket full of water and you go like this, you know, that centrifugal force that is pushing the water so it doesn't fall down, that's called free fall. And that's what we attain at low Earth orbit, microgravity, free fall. So what happens when these model microgravity experiments are used to further support the effects of microgravity on microbes in space? So we have this. This is one of the research investigations that served me to do my own research, because they set the stage for many of us who are in the space life sciences investigations. So what Nickerson did back then in the 2000 and 2007, she had a ground control, which means a ground control of salmonella. And then she infected mice with it. And then she had also another batch of salmonella, which became astronauts, so to speak. They went into space flight, and then they came back, and then they studied those samples. And so they looked a bit different, also under the microscope. There was this extracellular matrix, and you guess right, this is some biofilm that is formed here. And it just so happens that these samples were also injected into mice in the same conditions as the ground, but they were much more virulent. So this is where this situation comes, saying that, you know, bacteria become more virulent, in particular salmonella. And so in their manuscripts, in their publications, they went ahead and established that they claimed that because of their studies, it appears that salmonella has all these different genes, about 100 genes, that are involved in virulence, and they have been upregulated, and so to speak, they have been awakened, right? Once they have been in these microgravity conditions. Transcription enzymes have also expressed much more. They have been synthesized in a wider abundance, and iron regulation has also been upregulated, as well as the LPS. Now, LPS, for those of you who have microbiology training, would know that this particular carbohydrate and lipid together, it's very important for virulence for gram-negative bacteria, right? So this regulation, it's up-regulated, so to speak, when you're in microgravity conditions, right, for the bacterium. So that's that. And then, not only that, also in 2023, there was this other study saying that genetic exchange using plasmids in microbes is not really affected. Plasmids, by the way, is just like another way to store genetic information outside from the nucleus. And so it's not affected to genetic exchange, either in microgravity or model microgravity. So what does that mean if it's not really affected, you would think? So many things can happen. Microbes can communicate much better, right? So they can adapt much better, and the antibiotic resistance is increased. Okay, so now we get into the stage of my research, which had been in Bozeman, Montana. And there I'm just showing with that arrow, that was the laboratory I was working. And this is where I worked with Pseudomonas aeruginosa. And I was working with model microgravity, trying to understand, just like Nickerson, I was trying to understand for salmonella, I was trying to understand the same for Pseudomonas aeruginosa. So Pseudomonas aeruginosa is an opportunistic pathogen, meaning that if you encounter it, which is pretty much abundant in water, for example, in the toilet, in the sinks, much more so in the hospital sinks and the such, nothing would happen to you if you're healthy, right? And your immune system is in good standing. But if you are sick, if you're immunosuppressed, such as the astronauts, right, when they fly up there, then that's like party time for Pseudomonas, right? It's just the very good conditions for the Pseudomonas to infect and go about the infection process, right? So that's why it's an important organism to study. So what did I find? So in 2005, so I published, and I found that this particular microbacteria, so if you go ahead and subject that to model microgravity conditions, I wanted to see if virulence was upregulated or not. So we have to target what part of that Pseudomonas we'll be studying, and that was exotoxin A. This one is a protein that you'll find in Pseudomonas that accounts for is virulence, right? And so here I want to show you, this was the vessel. Once again, this is the model microgravity equipment or the high aspiration vessel. And once again, we're encountering here that this figure 2A, this is the microgravity orientation, simulating microgravity, in other words, right? And then the other one at the bottom, that's the normal gravity, the control position. And so the bacteria were subjected to this, right? But the bacteria, to be specific, they were inoculated or injected into these vessels through these openings, right? And these are just pebbles or beads, I'm sorry, that just to show, but this is not the actual bacterium. The actual bacteria looks different. It looks like this. So I took out the disc so that I can show it in the picture, but it came from a microgravity exposure, right? The model microgravity. This is a suspension of Pseudomonas aeruginosa. It looks greenish because of a particular pigment. So for those of you who have been very keen in studying your microbiology, you would say that's piocyanin, right? Piocyanin, it is a very good display of infectivity too of the Pseudomonas if it's getting a bit aggressive, so to speak, right? And this is another show. And then it's true too that in this state, because this is the gravity control position, you can see the green as well. But you cannot really tell much. How can you measure this? This is just for observation, right? But then you would take some of these and then you will look into the proteomics of it, which means the protein expression for that particular virulent factor, which is exotoxin A. So for that matter, in order to compare it to Nickerson's work with Salmonella, I hypothesized then that there is a similar effect from microgravity, right? From model microgravity. And I would go ahead and do this analysis, the proteomic analysis. That's what I did back then. And so I have different gels that I obtained, and in different types of situations, so to speak, right? So I started with the static conditions, like no movement at all, 12-hour and 24-hour, and those dots that you see on those gels, those are proteins that were expressed in those conditions. And then I went ahead and did the control position, horizontal position, or the earth conditions, and those are the proteins. You can see there tiny bit more that are being expressed. And then finally, I went to see after 12 hours and 24 hours in the model microgravity conditions, which are the top expression. So there seems to be a change, right? What does all this mean? So it just basically means that if there are environmental changes, there's going to be a virulence regulation of different kind, right? And so it might mean that microgravity existence there, that if you're exposing those microbes to, they give them an increased capacity to cause an infection. Okay, so then I move, then from there, I did more work I had done work, I'm sorry, after being there in Montana State University, I had come also from working with Dr. Marion Lewis. I had mentioned that I had worked with her with the space shuttle too, that had flown the Jerkett cells. But as far as the microbial work was concerned, I started in Montana State University, and then I went on to Dublin, the University of Dublin, Trinity College in Ireland. Dr. Charles Dorman was interested in my work with Dr. Barry Powell in Montana. So he contacted me and he asked me if I was interested in doing, in exposing or subjective salmonella to extreme conditions, right? And I say, yes, I'm the person to come to, right? To do all this, to bring havoc to bacteria. I just want to know what they're going to do. And so I went ahead and did my PhD there and studying salmonella in this case. And I subjected it to very low oxygen conditions. It had nothing to do with microgravity, but it was an extreme condition too. So I did my doctoral thesis on that. And so it was about this particular protein that had to do with virulence. It was, as it turned out, it was very well expressed. The expression was increased in very low oxygen conditions. So again, when there's extreme conditions, bacteria, in this case, salmonella, went ahead, not only for microgravity, like Nickerson had shown, but also with low oxygen conditions, it would become more virulent. So that's pretty much what I had just said here. And this is just a micrograph taken from the internet, how salmonella can change its environment to make it less aerobic or less aerated so that it can thrive, because it's a facultative type of microbe. Okay. This is just more on this research that had been done up in space so that you can have an idea. There you go again. Pseudomonas aeruginosa, salmonella, and even the fungi, Candida albicans. Okay, this was back in 2006. There's this one done in over time in the International Space Station, but only all the results and the analysis and the such was published back in 2024 last year. And this particular bacterium, I mean, it showed all this multi-drug resistance. And so it was very interesting to see that happening, right? How do they do that and how they follow that, right? They follow that resistance and how this transfer happened within the bacterium and the such. They found this. They found that there's all these happening in this particular bacterium. As you can see, there's the resistance. There's 13 strains. They found 13 strains having resistance to different drugs, and that's only because these genes, the beta-lactamases, had been altered. The same for the vancomycin resistance one. So this is, exactly consistent with what I had been saying before, right? So they are subjected to extreme environments, and then the microbes are going to respond in an aggressive way, so to speak, right? Okay. So all this paves the way for us to circumvent to find ways to circumvent antibiotic resistance or find non-antibiotic treatments solutions. So this, I find it very funny because they don't have to train, right? They're ready to go. They know how to thrive up in space, this bacteria. Okay, so where people go, microbes follow, right? So this is just historical things that happened since the 1980s. We had the space station called Peace or Mir for Russian, and then to the present, which is not going to be too long before it comes down, but more things, very exciting projects are there to go to Mars or to also continue the Artemis mission, right? So we still are learning from all these. Just remember what I had said about the water pipelines here. That's a real problem. Okay, this is just to let you know that in the School of Medicine, we are working very hard to show the students the importance of microbiology, of course, in medicine, but not only that, also that it's an emerging field also for space program, right? Because it's very important. I cannot emphasize it enough. And not only that, I mean, we have a SpaceX around the corner, so who knows? There's something on the horizon for us there that it's very interesting, not only for scientists, but also for physicians as we get into our space medicine too. understanding how astronauts respond, right, in their bodies, and also their interaction with the microbiome up there. This is current research that I'm doing with my students here. It has to do with the oral microbiome and also the brain one, and what could be the link between the two. And then there's the names of them. Is Noah, Taha, Dr. Quinones, who's another faculty, together with Dr. JC, another faculty here, and myself. We all have worked very hard together to understand these. Right now, we have been doing literature search, and later on, hopefully, we get into the wet lab of things. But this is our current work as it goes here, understanding these axes, and then the bacteria that have to do with these neuroinflammation, the red complex bacteria, neuroinflammation, and if indeed there's that magnetic link, which would help us control biofilm and the virulence in this bacteria or microbes in general, and how would this translate into the space microbiology, right? Understanding how this plays a role and how can we manage that up in space. So this is something that our students here in this team have achieved. I'm very happy, very proud of them. And they won in this symposium, this conference, research conference for best undergraduate and graduate oral presentations, talking about what we've done. And also I have been interviewed by NASA Solar System Ambassador, Dr. Thomas Spencer. If you're interested, you can always go to there. There's the link to understand more of what I've done, what I have done with other people and with students. And if you have any questions about how to embark into the space program, space life sciences. All right, so this is about antibiotic resistance. I won't go too much into it in the interest of time. I will just talk things that I think I've mentioned before, but to remember, not to forget the fact that astronauts get sick more often than not because of all the situations I've mentioned before. And just for you to have an idea, look at this. The survey of space shuttle missions from STS-1 all the way to 89 indicated infectious disease accounted for 1.4 of all medical events. And that's not including skin and subcutaneous tissues, okay? So that's pretty high. And if you think about, the small place they are in, and that is increasing, right? And then because they didn't even account for skin. So we have a lot to study here. And you need to remember that the closed environment, like I said, is so small, it's isolation, there's the stress, there's vapors and chemical. So all this is compromised. They recycle the water, the urine, the air, there's the radiation, there's dehydration. So it's everything like against them, right? But it's still worth it to be up there for many reasons, right? There's so much we need to find. So what to do? There's prophylaxis, there's vaccination. We need to track those opportunistic pathogens. That we can sterilize food to a certain extent, right? And we need to look into those air filters. We need to monitor that microbiome. So like right now, we just, our group, space team, as I call it, we had applied for a grant to study the space microbiome. And we're still waiting to hear from them, but we're hopeful. Okay. And we want to also study non-antibiotic approaches since they're very clever, they're very fast to resist antibiotics up there. So there's more on these, how to circumvent antibiotic resistance, exercise, deep clinic procedures, mental health strategies. They're having antimicrobial coating experiments in the space station. And this is just to almost done here. They are astronauts Sonny Williams and Butch Wilmot. They returned safely last March 18. Welcome back. Yay, we were like waiting for them for the longest time. And I just want to say that Sunita, she experienced some things, right? Being up there, she experienced what is called SANS. So the body goes through a lot of things, and this is pertaining to space medicine once again. So yeah, the brain, the eyes were affected, changes in vision. And so this is important to keep it in mind, all these things that astronauts have to go through, besides, of course, the big issue of the microbes. Okay, so takeaway points to finish up is that microbes are highly adaptable and resilient. They're able to survive in space by modifying their genetic framework. They are prevalent in spacecrafts and space stations. They are capable of causing a much more lethal disease than here on Earth and to astronauts because of their weak immune system. And so there's a need to continue these investigations to monitor through these microbiome studies, biofilm formation, drug resistance, vaccine potential in space, and also in simulated microgravity conditions. So all these altogether is going to provide us clue how to survive and thrive in space and how to sustain interplanetary travel eventually, and may hold the answers for the origin of life, right? Life origins. If you're hungry for more, I provide some of these. I don't know if you want to take any screenshots or any pictures. These are very good, I think, if you want to know more about astromicrobiology. Just remember, somewhere something incredible is waiting to be known, as Carl Sagan said. And it doesn't matter that we don't have all the answers. The quest to unravel the mysteries can be our greatest inspiration. This is from astronaut Tom Bazos. Thank you. These are my 3 kids, Leo, Juno, and Ethan. This is Leo, Juno, and Ethan, and my 4 dogs, Luna, Vanilla, Cookie Dough, and Peanut Butter. Thank you.
Thank you so much, Dr. Guadarrama. It was a really nice presentation. Interesting to know how the microbes are affected by microgravity, and especially they become more virulent. So, we have some questions for you. So,
okay.
I want to start with the first one. It said, have you found that there are any similarities between space microbiology and other forms of microbiology like marine biology, etc.?
Yeah, well, I mean, I guess the question comes or it's trying to tell me about the microbes that were found in the space station and marine microbes, perhaps?
Yes. Marine or earth or the thermophilus.
Thermophilus. Well, so far, it's not that we have found anything in the only similarity is that that we have found is the resilience, right? The resilience to even in these conditions that are altered in gravity, they're able to thrive. But as far as, for example, the pore formation that has been modified in some of these extremophiles that can live under very high pressures or like the gamma tolerance, as far as we know, well, there's a lot of studies there, but there's much to be done to understand that yet, because it's not that they have sent, for example, this to space, like out there, and retrieve microbes and see if they can survive. I have to say, though, that there's this controversial thing that back in the Apollo mission, because they were using too many cameras and some of them were left there between every mission throughout the decade of the 60s, they retrieved a bacterium, supposedly, that had withstand the harsh environment of the moon. But this is still controversial because they say that no, that came from one of the astronauts and whatnot. But I wouldn't be surprised that the spore formation or the spore bacteria, the spore can actually withstand the harsh environment of the moon or outside the space shuttle, not the space shuttle, but the spacecraft or the International Space Station. As far as the marine or the air microbes, that we're still looking. I don't know if that answers the question.
Yeah, thank you. Another one is talking about how the microbes, when they're in the space, they become more virulent. Is how this space microbiology research can help us understand how Earth-based disease.
Yes. If it would help us understand much more disease processes?
Yes.
Yes, because, for example, like salmonella, like understanding that if the gravity vector would be a big thing for it, what is the makeup of what are the genes that the bacterium tweaks, right, when it's encountered in a different environment. Now, the gut environment, for one, is that of fluidity, right? There's fluids. There's not necessarily high shear, but it could be low shear, just like in microgravity, the same as for the oxygen conditions, it could be very low. So it let us understand much more how the salmonella, for one, goes about in the disease process, because we're putting it in an environment that is very similar to the gut, for example, right? And so it's like having like a first row view of the spectacle of the disease process of this particular bacterium. And it's not the only one. I mean, there's many other in which we can understand what would happen if we put it in another environment. And if it would be better, if it would be worse. But it's very unlikely that once all this happens up there, that when they come back to Earth, they will behave the same way, because the environment is different. So there's so many different factors to account for.
Okay, thank you. In one of the pictures, with you taking the samples of the special, remember you were wearing a special attire? They were
Yeah.
asking if you wearing that because of the unknown microbes that you might encounter, and then maybe they cause a disease in contact with the humans.
Yes, very good question. So it's both. It works both ways, right? We didn't want to plant any microbes if we could reduce that, because, like I said, there's no such thing as 100% sterile. If we can prevent bringing anything from outside, because if we put something there that might get the astronauts sick, or we don't want to get anything back. So it works both ways, right? So that's why we have to be in that special attire, because once they get back, they have to be quarantined. The astronauts, they need to be quarantined before and after for the same reasons.
Okay. So another question is, what has been the most surprising, unexpected finding from your work in space microbiology?
What has been the most surprising?
Surprising,
Unexpected
finding
finding.
in your research.
Well, that's a good one. Well, for one, I didn't expect that to see that microbes would become more effective, because we tend to think that, right, like all living organisms, they share the same attributes, right? And so if we are placed, for example, in space, we don't become super powerful, right? We don't become Superman or Superwoman. And so seeing that bacteria, microbes become that, it could be a bit scary. But in order to circumvent that fear in myself, just like Marie Curie used to say, we tend to understand. Once we understand, we are less fearful, right? So that was the big thing for me, seeing that they become much more infectious, and they're ready to do whatever it's needed to propagate life, but in their own terms. So that's the most fascinating thing for me. Oh, I cannot hear. I cannot hear you. I cannot hear you.
Okay, I'm sorry.
Oh.
Do you have any advice for a student who wants to work in NASA?
Yeah, so we are living very interesting times, and we are very fortunate. So Texas in particular, because there's many, not only NASA, there's many different space companies that are rising like left and right. And there's this new institution, or this new body of knowledge that is called the Texas Space Commission, that it's newly formed, and they have much funding to give for space research, space life science, space health, space aerospace engineering, all this, because they want, and this was because of Governor Abbott, who decided, okay, we need to become the #1 hub for the space program. So if we're here, you already, if you're interested in the space program, you already have done, like, checked the first step. You're in the right state. And so the next step would be, yes, NASA, if you like to be part of NASA. And there's many different internships that you can do right now at your age, if you're an undergraduate. If you're a high schooler, there's others that are being sponsored here by UTRGB. I can get you in touch with the people who are doing those type of programs, to get you, like, you can have your right foot in with NASA, doing those internships. Doesn't matter in what field, because there's many different fields. It's not only engineering, right? There's also the life sciences part, other than that engineering. And so, and then there's a SpaceX, and there's Blue Origin, there's Galactic, there's many different things that could happen there. But I can put you in contact with them, but we have to understand what do you want to study, because there's so much out there that we have to look to see where do you want to go. Now, if you are interested in research in the higher education, in academics here, we can start either here with me or with the Texas Space Institute that is part of UTRGB2, where one of my colleagues, Dr. Campo Casas, he works with astrobiology. So his work has to do with the microbiology, or the life that it's already out there, right? Interplanetary space. Mine has to do with terrestrial microbes and how do they behave in space. So we kind of complement each other.
Okay. And the last one, still talking about NASA. What was your favorite team when you're working out there? And do you have faced any challenges?
My favorite team?
Favorite thing that you do in there in NASA?
Oh, favorite thing. My favorite thing. Well, for sure, it was when I was able to get inside the space shuttle and be there with, also working with astronauts and speak to them outside the shuttle. Of course, it was not like other people who had the opportunity to get them ready and whatnot before lunch, but it's just understanding how all that environment works and all the different things that they do, right? I mean, the training and the way they see life, and the way it's a different kind of thing, because it gives you perspective, right? Like what's really important, that we're very fortunate to live on this planet, and that there's still so much to learn and to understand from microbes, for sure. But not only that, life as a whole, right? And then there's, like I said, like Dr. Chang Diaz, who's an astrophysicist. He has so much knowledge how to do interplanetary travel, or to work with Roger Crouch here, that is in the picture that I have right here, that I met him. Very kind people, very focused, goal-oriented. So, I mean, you learn from everyone. And I think that it's another important factor, the teamwork, right? That you cannot do anything alone. The teamwork part too, it's very important.
Okay, well, thank you again for your time and for sharing your expertise with us, and also for giving us a glimpse of what happened with the microbes in space.
Thank you. Thank you for the invitation. And please do contact me if you have any other questions. My email is there on the screen.